Hello students, in today's video we'll discuss about the DNA, which is deoxyribonucleic acid, which is deoxyribonucleic acid. So if we talk about the DNA, so DNA is present in the cell. For example, this is your cell and this will be a nucleus of your cell. Inside the nucleus there will be a genetic information which tells you about yourself, that who are you. So to answer this question we should familiar with this one, and if we zoom in, then inside the nucleus we have genetic information in the form of chromosomes. So if we talk about the DNA or the chromosomes or the gene, we all know that every human or individual have 23 pairs of chromosomes. How many pairs? 23 pairs of chromosomes. 23 pairs of chromosomes. Let's talk about the chromosome structure that we have. This is a central point which is known as centromere, and we have also chromatids which are like the arms. We have P arm and we have Q arm. The central point is known as centromere, as I've discussed. So this is a chromosome, but this is also a chromosome. The difference between these two chromosomes, that this is a chromosome which is present before replication, that during the cell division this chromosome will get divided, will get replicated to form this chromosome. So whenever the cell divides, the chromosome or the DNA, the genetic material, will divide itself also. So that's why the chromosome before replication, and this will be a chromosome after replication. So P arm and Q arm, as I discussed earlier. So but how it is possible that this small nucleus, this is a very small nucleus, you can see this is a very small nucleus, that how this a small nucleus can accommodate these 23 pairs of chromosome? This is all due to the wrapping power, means how DNA is wrapping itself around the histone protein. This will decide that these are the histone protein around which the DNA will wrap itself, and when the DNA will wrap itself around the histone protein, then it would be able to fit itself in the very small nucleus, because we have an estimated size of the DNA is like from sun to earth. So that's a huge gap between the sun and earth. But if we say that this small nucleus is accommodating such a very large genetic material, so whenever the DNA will wrap around the histone protein, it will get set, and it will accommodate itself, and this will form the proper chromosome structure. You can see this is a DNA. If we zoom in, if we can see that this is a DNA, and if we zoom in this structure, you can say that the DNA will be present like this. So this is a DNA. You can see this is a DNA. Inside the DNA we have these bases. What are these bases? This is like a backbone of the DNA. This is like the backbone of the DNA, and this are the bases. If we talk about the normal DNA structure, we have three components. How many components? We have three components. One of them is the bases. I have written earlier, I have informed you earlier. The second component is the ribose, the sugar molecule, or the ribose sugar in case of the RNA, or deoxyribose sugar in the case of DNA. Deoxyribose sugar in the case of DNA, and the ribose sugar in the case of RNA. And the third component will be the phosphate group. This phosphate group is negatively charged, so that's why our DNA is the negatively charged. Is this clear? So next point we will discuss in the DNA is genes. In the DNA is genes. So if we talk about the genes, we know that gene is a part of DNA which will carry your traits, So what are these lines? These lines are the bases. We have four bases in the DNA. One is adenine, thymine, cytosine, and guanine. The adenine will always pair itself with the thymine, and the cytosine will always pair itself with the guanine. So these are the bases which are present in the DNA, while in the case of RNA we have uracil instead of thymine. So is this clear about the DNA or the RNA? Now it's time to get into the genes. Whenever we talk about a very little bit or a small portion of DNA, for example, we have just taken this portion of DNA, I am calling it a gene. I am calling it a gene. But keep in mind that every portion of the DNA can't be a gene. If we're talking about the gene, then gene is the only that portion which will carry information for the transcription, means the only portion which will get transcribed. For example, this is your entire DNA, and if only this portion of your DNA is converting itself into messenger RNA, for example, this is your DNA, and if only this portion of your DNA is converting itself into messenger RNA, then this portion is known as gene. Those portions of the DNA which are unable to transform their self into messenger RNA, that portion will not be a gene. Gene is the only that portion of the DNA which will transform itself into the messenger RNA, or the process through which the DNA will convert itself into messenger RNA. That process will known as the transcription. So with the help of transcription, the DNA will convert into the messenger RNA. So that is a very brief overview about the DNA, about the genes, about the chromosomes. So if we want to learn more about this, so stay tuned and updated.